It is often said that science is, even fascinating than fictions that we have read through. And I think the perfect example, one more perfect example of this particular proverb is the development of the first homegrown vaccine of India, that is Covaxin. And there is a lot of story behind the development of the Covaxin, and one of the major vital part that has been played in the development of Covaxin is by National Institute of Virology. And we are really happy to have the leader of National Institute of Virology as a speaker for this particular day, Professor Dr. Priya Abraham. Ma'am, please come to the stage. Please put your hand together to welcome Professor Dr. Priya Abraham. Now I have the honor and privilege to welcome on stage Director, Headquarters, National Council of Science Museum, Sri Samrendra Kumar sir. Please put your hand together to welcome Sri Samrendra Kumar. We would like to welcome our speaker for this particular event, Professor Dr. Priya Abraham, with a flower bouquet. I would now request Director, Headquarters, Sri Samrendra Kumar sir, to please welcome our speaker with a Uttariya. Now I would request Director Science City, Sri Anurag Kumar, to please welcome her with a memento of 25 years of celebration of Science City. Thank you, everyone. Now it is my privilege to invite Director, Headquarters, Sri Samrendra Kumar sir, to give his formal welcome address and introduce the speaker to the House. Thank you.
So, a very good morning to one and all present here in this auditorium. Distinguished guest for today's event, Professor Dr. Priya Abraham, Director, National Institute of Virology, Pune. Distinguished guests, ladies and gentlemen, present in the auditorium. My dear young students and the future scientists of India, friends from media and colleagues from NCSM present in this auditorium. It's a great privilege for me to give a warm welcome to all of you here for this very interesting and exciting lecture to be given by Dr. Priya Abraham today. In fact, we met Dr. Priya Abraham personally in Pune, just to inform to all of you that National Council of Science Museum is basically developing an exhibition called Vaccines Injecting Hope. And that exhibition is going to open in November 2022. And in the process, we contacted several institutions, and NIV is one of them. And we got a very tremendous support from Dr. Priya Abraham, and that story is so fascinating, which you will hear from her very soon. So I think it's wonderful to, and she's an excellent TED speaker also. So I think you will all enjoy this her lecture. So it's basically it's a privilege from me on behalf of Science City to welcome Dr. Priya Abraham once again and all of you present here. So let me just say a few words about Dr. Priya Abraham, because She had completed her MD and PhD in biology at CMC Vellore and as an illustrious career studying viral infections. She is a medical doctor and teacher who became interested in infectious diseases, especially diseases caused by viruses. She was head of the Department of Virology in CMC Vellore in Tamil Nadu, and in November 2019, she took up the responsibility of the director of National Institute of Virology at Pune, Maharashtra, just before pandemic. She and her team discovered the first cases of SARS-CoV-2 for the first time in India. They achieved many firsts in the battle against COVID-19 and finally teamed up with the Bharat Biotech International Limited to make India's first indigenous vaccine, which is called Covaxin now. She has been given, I think, numerous awards, and I think I should not, but some of them are very exciting. That professor was awarded Business Excellence and Innovative Best Practices Academia Award 2020 by the New Delhi Institute of Management for her extraordinary achievements and exemplary contribution to the society, and also conferred the prestigious Integral Health and Wellbeing Council ISW Gyani Awards for Medical Research Leadership 2021. More recently, she got the Rotary Vocational Excellence Award, and she has been on WHO panel for so many things, and probably much more she can explain when she talks about in the lecture. So here I invite Professor Dr. Abraham here to tell the story of NIV's, National Institute of Virology's multi-pronged effort to fight this pandemic and the discovery of this Covaxin. Thank you.
Am I audible? Okay. So, respected director and director of this institute of the NCSM, which is part of the Ministry of Culture, other respected staff of this institute, members of the press, and my young dear friends. I hope I will be doing justice for this honor that has been bestowed on me to speak to you about, I think this works better, speak to you about the journey that the ICMR-National Institute of Virology undertook from the start of the pandemic. To be a little anecdotal, you know, when I attended my interview in late 2019, at the interview I was asked, if there is an outbreak in the country, what will you do? Well, I did answer very spontaneously, but I bagged the job. And it was almost as though it was, tailor-made for me. Within 2 months, we were on the threshold of a pandemic that still continues today. So I will just take you through some of the efforts of National Institute of Virology. It was a roller coaster ride. We were asked to do many different things. Of course, best known is our contribution to India's first indigenous vaccine. But I'm here to tell you how, if you're willing to pitch in, there is work to do in our country. And that's what my colleagues at the National Institute of Virology did. And that's how we were able to deliver to our nation in so many, many ways. So way back in, I think I'll take this off. I'll be audible on this mic, I think. Excuse me. Way back in December of 2019, when there were talks and media reports of this unusual lung infection, which we call pneumonia. Inflammation of the lung is known as pneumonia. That was being reported in China. I got together with my physician scientists, doctors, medical doctors who have become scientists. And I said, we need to look at this carefully. Cases are multiplying. We need to study the presentation of this unusual pneumonia and, get prepared. I also met with the laboratory scientists who have a very strong laboratory training, and I told them we need to put together detection tools so that should this infection come to India, we should be prepared. This was in December. In January, we had more detailed discussions and both teams got together. The medical team made these, patient request form, giving details if a patient has certain symptoms, what we record when we send a sample to a hospital. And what you're seeing there is actually some of those patient request forms that we made way back in January 2020. And this is me sitting with some of my colleagues here. And the laboratory scientists also put together a real-time PCR, which many of you know as the RT-PCR, to look for this new coronavirus, whose details were by then submitted to WHO. So this was our way in being proactive. We were watching the news. We were listening that this is becoming an expanding problem. The previous coronavirus, known as the Sars virus, which broke out in 2002, it spread through the world in a flash of a few days because of increased air travel. So we knew this too is likely to spread to different parts of the world, and that was why we took this proactive measure. So we put together a RT-PCR and we sent it to all the key laboratories, government laboratories that were closest to major airports. We have a network in the ICMR known as Virus Research Diagnostic Labs, VRDLs, and we have about over 136 of them, but we sent it to 13 such labs that were closest to major airports. Again, for the reason that I just stated, we knew infection could be imported very easily in this day of increased air travel. We sent them not only the reagents to do an RT-PCR, we sent them the standard operating procedure, what you call the recipe to do the RT-PCR. And we also send them positive controls, because when they run the RT-PCR, if they get negativity, is it because the test that they are running is good or bad? But if you have a positive control and that positive control is coming positive as we expect it to do, then we know our RT-PCR is working. So we send them out, and we didn't just send them out reagents. We were not able to travel easily in those days, so most of the training was online. We are now in the era of training online. So we had training sessions, hand-holding sessions, troubleshooting sessions that went on 7 days a week, and we worked 7 days a week up to August 2020, training more and more labs. And today we have 60 to 70%, over 1,300 labs in the country that use the RT-PCR that we developed. So when we did this training and capacity building, lo and behold, on January 30th, actually 29th night, we got our first positives. I was in Delhi. I was returning to Pune. I was approaching the airline gate to board the flight, and I got a call saying, "Ma'am, I think we've got a couple of positives." So I said, "Hold on, I'm coming to also look at your results." And what is very important is we should not be gung-ho and over-enthusiastic to report as a result till we have confirmed it. So what I had already requested my colleagues to do is to work as a team two, team A or team A, team B. Team A did the primary testing. Team B got those samples coded. They didn't know which was positive, which was negative. Team B was asked to run the test. And when Team B got exactly the results of Team A, I knew we were in business, that we had the first SARS-CoV-2 cases. So I reached Pune by 10:30 p.m. And with me was a very senior official from ICMR, but I didn't let him know. He was my immediate boss. I wanted to confirm it myself. So I went to the lab, reached around 11-ish, looked at results of Lab A, Team A, and yes, that was suggestive of a positive. It was very late at night, and Team B was in the other campus. So next day morning, I went home by around 2 a.m. 6 a.m. I was in the other lab and checked their results. The results concurred 100%. So then I reported it to my immediate boss, who then went on to tell the Director General of ICMR, and then the rest is history. So the one lesson learned from this is, when we embark on something new, sensationalism is not the key. We have to be absolutely sure when we report something that has so much gravity and importance to the country, that we actually be very, very sure of our results before we say this virus, this COVID-19 virus, SARS-CoV-2, has actually come into our own country. So that was the beginning, and now I'll go a little faster. So these are pictures of lady scientists and technologists, and one of the gentlemen who were putting the reagents together. And remember, when we send out reagents, we just can't pack something and send it away. It should be of quality. So this was, to me, a sacred job, that what we send out has to be of absolutely supreme quality, so that whichever lab receives it to do more RT-PCR, we should be spot on. So this was the activity, and we became a factory of sorts. Our garage, as you can see some cars in the background, the garage became a place where we put hundreds and hundreds of boxes. We threw in dry ice, because dry ice preserves the reagents, preventing it from decay so that the RT-PCR will work optimally. And we packed it up, sent it out even by the latest flight out of Pune, or the earliest flight. We continued this even during the lockdown. And as of today, we have sent over 64 lakh RT-PCR reactions to different corners of the country to enhance and equip the laboratory testing in our nation. Now, when we send out material, like I referred to quality, who is to test our quality? We are sending material to other labs, assuming we are of a good quality, but we need to be assessed on an international platform. And so we engaged with the World Health Organization. They have something known as an external quality assurance panel. So they sent us this coded panel. We tested and gave them the results. And I'm happy to say, on 3 such occasions, we have got 100% score. So that means our testing is spot on, and we are equipped to truly be a provider of good RT-PCR reagents and kits for the rest of our country. This was one of kit that we made. This is actually reagents made in NIV. We made the packaging, even the logo, everything was, we put thought into it. We miniaturized this kit so that we could almost quadruple the number of tests that we could run. And today we run a quality assurance program for the nation, getting WHO external quality panels, and we have at least 1,300 government and private labs using these quality control sera so that they can check their own quality, and that is a cornerstone of testing. When we do a test, it should not be a result, it should be the result. We should be spot on. So we continue that even today. Oops, I don't know what I did. Okay, and we also did, I'll just go back we also did something which was very important for the nation. By around April, May of 2020, the whole world had increased their testing. So reagents and what we known as RNA extraction kits, they were all running dry. We did not have any reagents. Even the nations of the developed and richer countries were running dry of material. So a number of companies in India came forward with their own formulations of RT-PCR kits, RNA extraction kits, viral transport media. Many of you would have seen those swabs being taken and put into a tube. That tube contains something known as virus transport medium, which keeps the virus healthy and alive so that we can do the RT-PCR. So number of companies came forward. You can see here, I'm so sorry, yeah, you can see here a number of companies came forward to provide their material, and only some of them passed the test, but even passing that many tests was very important because that gave us a self-reliance. The PM's Atmanirbhar initiative was also implemented in National Institute of Virology. So when we said an indigenous kit was passing our validation results, that became our own capacity, not depending on other countries, to continue our RT-PCR testing. Today, we have also validated some kits which can specifically detect the latest variant of SARS-CoV-2, namely Omicron, which many of you have heard about. So our next achievement, within 2 weeks of getting the first SARS-CoV-2 cases, was to visualize this virus under the electron microscope. I have taken actually another image because this is a little clearer for you as compared to this. This requires a little more specialist viewing. This will explain to you that when you look under the electron microscope that amplifies your magnification over 100,000 times, what you actually see is the body of the virus with these spike-like projections. Can you see? Can you see these spikes all around the virus? These are different images of SARS-CoV-2 with a fringe of spikes which resemble the spikes or the protrusions of a crown. That's why it's called coronavirus. Some people also refer to it as the rays of the sun. And that's where this name came about for this family of viruses, those spikes that stick out of the virus. We were also the first to detect the whole genome sequences of the virus. Now, when we talk about whole genome sequences, you know we all have our own genetic code, so do viruses. Viruses can be either RNA viruses or DNA viruses. This is an RNA virus. So we mapped the full genetic code of the virus. It has 30,000 nucleotides. And I always give the example of the beads in a chain. So when you want to look at what is the makeup of individual beads, you know RNA and DNA has 4 nucleotides. Imagine it's a chain with beads of 4 colors. And those sequences of the colors of the beads can change, which can change the genetic sequence of the virus. So when we looked at the genetic sequence of the virus, 30,000 nucleotides long, we published the first 2 sequences from the country, from the National Institute of Virology. Now I'll go on to another job that we did. I need to tell you a little about viruses before we talk about isolating the virus. Now the virus is a microorganism that is described as an obligate, that means compulsorily, an obligate intracellular parasite. It cannot grow in an inert medium or in a flask full of good reagents, no. It has to grow inside a cell. And this is an image of the virus entering a cell. Okay, it attaches to the cell's receptors, okay, and then it gets inside the cell, slowly uncoats itself, releases its virus genome, and using the cell's own machinery and its own enzymes will make more viruses of its own. So in a sense, it hijacks the cell's own machinery to very selfishly make more and more virus particles, which are eventually released by the infected cell to infect more cells. This is what viruses do. And to grow viruses artificially in vitro, we have what is known as, this is a tissue culture flask. And on the base of this flask is a single layer of cells, what we call a monolayer. This may be of animal origin or human origin, and we grow a single layer of cells. What you're seeing, that red material on top, is nutritive medium for those cells to stay alive. And into that flask, if you add infected material, and if the virus recognizes the receptors on those cells, the virus goes in and produces certain changes in the cell line, which are known as cytopathic effect. So to show you that, on your left of your screen is the single monolayer, which is not yet infected with the virus. This is a cell line which came from the kidney of African green monkeys. So this is known as a vero cell line, and this is an uninfected cell line, spindle-shaped cells growing on the side of that flask which I showed you. But when you put virus into it, what happens is the virus grows, the cells start coalescing, joining together to form giant cells. They look different, they get rounded, they look more refractile, and that is what you call as cytopathic effect in a vero cell line. Can you see the startling difference? Uninfected, infected. And this is a close-up of the infected cell line. You can see the rounding up the cells, cells joining together, forming a multinucleate cell here. And these are the changes that suggest we are successfully growing the virus artificially in a layer of cells. So the capacity to isolate and grow the virus artificially was a big, big achievement for National Institute of Virology. And this paved the way for the vaccine. I'll come to that. So we grew it up, and even there, when we grew, and my colleagues told me, ma'am, it looks like the virus is growing. We did not go to town about it because we confirmed it once again in the electron microscope. We did something known as immunofluorescence testing. We did again looking at the genetic makeup of whatever was growing in the cell line. When we confirmed this is SARS-CoV-2, then only we went public. So Jan 30th, we discovered the first cases. After a lot of persistent efforts, on March 12th, we met with success growing the virus, and we were the 5th country in the entire world to successfully do so, and for the first time in India at the National Institute of Virology. So this cannot be undertaken in an ordinary lab. It needs to be done in a lab of higher biocontainment. Why do we say biocontainment? Higher biocontainment labs are labs that are under negative pressure. That means anything from outside comes in, but nothing from inside goes out, because it should not contaminate the environment. When you work in a biosafety cabinet, any exhausted air is irradiated and zapped so that no infectious material gets into the environment. So those kind of labs are known as biosafety level labs 3 and 4. And this is an example of a biosafety level 4, the highest containment lab in the country, only one of its kind at the National Institute of Virology. And you will see this man looking little inflated. He is looking inflated because he's wearing a special paper suit, and this paper suit is under positive pressure. Why is it under positive pressure? It is sending air outwards from him, because should anything air get into his suit, it should go away from him. So this kind of facility protects the individual who's working in the lab and also protects the environment. Lab in negative pressure, individual wearing a positive pressure suit. And you can also see in this picture a lot of those flasks which I showed you, where we were growing it up, again, factory style. Lots and lots of virus was being propagated in the laboratory. This is the kind of lab that can handle the most dangerous pathogens in the world, be it Ebola, Nipah, Crimean Congo hemorrhagic fever, and so on. Coronavirus, SARS-CoV-2, requires only a biosafety level 3, but we were not taking chances because we were going to grow up lots and lots of the virus. So this happened in a biosafety level 4, the highest level of safety in the country. In the midst of all this, the government of India asked, through my boss in Delhi, that could National Institute of Virology send out a team to Iran in March 2020, because a lot of Indians were there in Iran, and Iran was having a particularly serious outbreak of SARS-CoV-2. There were deaths. If you go back into your YouTube or other social media, date back to March 2020, Iran, you will see a lot of body bags lined up like sardines because people were dying very rapidly. So the government of India felt that Indian nationals, be it students, tourists, pilgrims, what have you, needed to be repatriated, brought back into our country. And therefore, they wanted National Institute of Virology's team to go out. I was not very happy because that time there were huge fears about what SARS-CoV-2 could do, but duty calls. So I actually had to send out a team of 6 people, which was 2 scientists, an engineer, and 3 technical team. And I'm very proud to say, my colleagues here, shown here, did not for a moment show any hesitation. They said, "Ma'am, we will go." It brought tears to my eyes because I was very scared for them. But they said, "No, we will go, ma'am. We will go to Iran. We will screen individuals, and we will help in the repatriation of or bringing back our own citizens to our country." So these are pictures of them in Iran. They were working in a rather busy neighborhood in one of the cities. And different countries have different challenges. And in one particular area, they felt they were encountering some resistance. And they sent me messages to say, "Ma'am, the soldiers, the public are looking at us oddly. They're looking a bit hostile." So I said, "Just take all your testing material and come to the Indian embassy. Do your screening in the Indian embassy where you will be the safest." So in fact, these are pictures of them establishing themselves in the Indian embassy, and they tested over 2,000 individuals whose samples were sent by, it was a big diplomatic venture by the private airlines, the Indian Air Force, the Indian Embassy in Iran, and the Ministry of External Affairs all got together, sent the samples to us. They reached here at around 10:30 in the night. We started testing over 2,000 of them. By 9 a.m., we had to start giving reports of which samples were positive and negative. Negative people could immediately be shifted back. The positive individuals had to remain in isolation and then be brought back into our nation. We also held in the repatriation of another cruise ship. Many of you may recall, it was a cruise ship that was docked off the coast of Japan, the Diamond Princess cruise ship, one of those luxury cruise ships. We repatriated Indians to come back into our country. Likewise, from the Italy, where many individuals were dying, again, Indians were repatriated, and we did the same exercise. Samples came in at some odd hour in the night, and next day morning, they had to give the results to expedite their repatriation. We were also called to do lots of training. Apart from training all the government labs, which I told you about, we did some physical training, which was limited because the lockdown came into being. But we were very honored to train the NDRF, the National Disaster Response Force. The group that goes to the rescue of citizens, where there are natural disasters of earthquakes, floods, whatever. They wanted to know how to do coronavirus testing. And you can see some of them here in the picture here. We were training them. We trained many of the medical colleges in the state of Maharashtra. The rest was all online training, but we did limited physical training for some of them. ICMR, the headquarters in Delhi, said, प्लीज़ चलो आप लोग एक वीडियो बनाना है सभी को सिखाने के लिए हाउ टू डू SARS-CoV-2 testing in a very using good laboratory practices, using complete biosafety measures. So we made a video of all our testing and assembled it in a sequential manner. So anybody who was new to RT-PCR testing could pick up the cues. Because we were able to grow the virus, that empowered us to make the first antibody detection ELISA. ELISA expands as enzyme-linked immunosorbent assay. It's an antibody detection assay, and this was coined the name Kavach ELISA. And this was the first antibody detection in completely indigenous assay that we made. It was used for the first national sero survey and was also used to screen individuals before they took part in the clinical trials, both for Covaxin and for Covishield. So we made that. We also made, I won't go into, this is very technically demanding, but it's known as a neutralization assay, where you're looking at the level of antibody that an individual may have that can neutralize the virus. So it can go, the antibody goes and docks on to the receptors on top of the virus, and then the virus is not free to go and attach to the cells of the human body. So that is known as neutralize. You know, like we say, the soldiers neutralize the enemy's efforts. Like that, the antibodies can neutralize the virus's infective capacity. And so this is called a neutralization assay to know how much of neutralizing antibodies an individual has. This there was a big human convalescent plasma trial where at one time we thought taking serum from individuals who had recovered from SARS-CoV-2, we could take those serum, which is full of protective antibodies, and treat fresh people who had been infected with the virus. It was a big national study known as the Placid study, and in the Placid study, NIV was the only lab that measured the neutralizing antibody titers for this particular study to see the amount of protective or functional antibody. We improved on that assay, and this assay is particularly useful when you want to look at the vaccine response, antibody response to vaccine. It's known as the plaque reduction neutralization assay. I won't go into the techniques, but if anybody wants, at the end of the lecture, I can elaborate. But this is the gold standard of antibody testing when you want to check the amount of antibody, particularly after natural infection or after receiving a vaccine. So we developed that as well, completely indigenous. We had the craziest of demands. There were people who would ask us to check out creams, lotions, powders, all kinds of extracts, saying, आप देखिए इसका antiviral properties है. And we tested over 100 different compounds, of which we found about 22 had some antiviral property. And there was a lot of pressure, even from very powerful people, I should say even political pressure, to say, you know, test these compounds and these formulations because it has got antiviral properties. But we took a firm resolve to be scientists, to test it in the true scientific temperament. And yes, we could identify about 22 out of the 100 odd compounds that were delivered to us did show antiviral properties. We also were able to file some patents. That means we were able to make a unique RT-PCR. I think I showed you the image of that, which increased our throughput. It was again put together and assembled completely in National Institute of Virology. We put up another assay by one of our scientists in Mumbai. We have a field unit in Mumbai, Dr. Nandi, and he made something known as a field-adapted assay known as the RT-LAMP assay, and both these have been put up for patenting a patent has been filed, and the RT-LAMP assay has also been filed for international patenting. We also became a biorepository of different samples that came from infected people, be it throat swabs, nasal swabs, liquid from inside the depth of the lung. It's known as bronchoalveolar lavage, sputum, blood, urine, stool, you name it. All these clinical specimens that came from infected people were sent to our lab and to 2 of our field units, and we have them all stashed away in our freezers. But there are other labs in the country also that are doing this biorepository work. Another request we got was from companies that, you remember I told you about convalescent plasma or serum, where it is assumed that individuals who have recovered from infection will have lots of antibody, and that becomes a ready source of ready-made antibody to treat individuals who have COVID. So companies who make large-scale antibody in horses. So if you give the same antigen to horses and repeatedly vaccinate horses. Horses are large animals. You keep vaccinating them 10, 14 times, they will produce lots of antibody. And you take this much of their serum, it is loaded with antibody. So this is known as hyperimmune equine. Equine means horse, equine serum. So there were companies who came forward to say, we will make hyperimmune equine serum. So we gave it to 5 companies, and they took away our virus, which we inactivated for them, gave it to them. They gave it back to us for testing, and then they modified that antibody so that horse antiserum should not be reactogenic, should not cause allergy in humans. And then again we tested it and we estimated the titers and gave it to them. But this practice of giving ready-made antibody is not being received with that much acceptance, because human trials did not show good results. But at that time in 2020, companies came forward to make it in horses, and we gave it to them. Now, to give you an analogy, you must have heard of when somebody has a snake bite, you give them anti-snake venom. Anti-snake venom is actually a cocktail of high concentration of antibody, which is made in horses. Okay? So that same logic was used even to make hyperimmune anti-SARS-CoV-2 equine serum, and that was the genesis of this effort. And even Serum Institute of India came to us, and this picture depicts for you how many times the horses are vaccinated. And finally, the titer of antibodies runs into lakhs, because horses are large animals. You can give a lot of inactivated virus to them, inoculate them repeatedly, and they produce a very good immune response. Then came the start of the vaccine journey. We had isolated the virus on March 12th, and we had gone public about this. And lo and behold, knocking on our door on March 13th was Bharat Biotech International Limited. He wrote to me as well as to the Director General of ICMR, saying, we are interested in making a whole virus inactivated vaccine. That means the virus is kept whole. We are not taking out parts of the virus. The whole virus will be inactivated, zapped, and we will make a vaccine out of it. So I readily agreed, and of course my boss also was very enthusiastic about it. So in March, they came to us with the request to make a vaccine in partnership with us. So we had isolated the virus, we had grown up the strain, made sure the virus was growing very well in vitro inside those flasks which I showed you. And in end of April, we handed over the virus to Bharat Biotech. They inactivated it with a compound known as beta-propiolactone, BPL. They gave it back to us for testing. to check whether the virus was truly inactivated. We again tested its antigenic, immunological makeup, its genetic makeup, and then began the journey of preclinical trials. I'll tell you about it. I'll explain that in a while. And then the trials in human beings. So this, in a capsule, is the journey of Covaxin, India's first indigenous vaccine. So this is an image of two of my colleagues. This is Dr. Priya Yadav. This is Dr. Gajanan Sapkal. Her lab grew up the virus. He continues to do all the antibody tests. I showed you the very specialized antibody tests. They are handing over the live virus, packed in dry ice. They came in a vehicle. Lockdown was just about to be declared. They drove all the way back, carrying this living virus in this container, all the way back to Hyderabad. They grew it up, and then I had outlined to you the journey from then on. Oops. So to give you, there are different COVID vaccines, but to put it in context, we know we have the Covishield, which is widely given. Almost 85% of the vaccination given in our country is Covishield. It's a vector-based vaccine, adenovirus vector, which is carrying the gene for the spike protein. There are recombinant bacterial vectors. This has not yet gained great momentum. There are RNA-based vaccines. You know the companies. You must have read a lot about Pfizer and Moderna. The new vaccines, namely Covovax, Novavax, these are recombinant subunit vaccines. Then synthetic peptide, not yet gained great popularity, neither is live attenuated. And the last is the whole virion or whole virus inactivated vaccine. And you can see here that Covaxin, along with some Chinese make vaccines, are in this genre of COVID vaccines. So this is what we worked with. And we started our studies with the preclinical trials. I told you preclinical trials are nothing but trials that we do in animals. No virus candidate or bacterial vaccine candidate can go into human beings straight away. We need to first check the properties of generating a good immune response, safety and potency in an animal model first before we can take it into human beings. So our first animal model, of course, we had worked with some rats, mice and rabbits, but we did what is known as virus challenge studies in golden Syrian hamsters. And what you're seeing here is an image of the hamster. It's a very cute little animal that you can hold in the palm of your hand, okay? And this is the hamster. And what we did is, Bharat Biotech gave us 3 formulations of the vaccine, which had different concentrations of the virus antigen, as well as a compound known as the adjuvant. An adjuvant is a chemical which potentiates or augments or enhances the immune response or antibody response when we inject the killed virus antigen into a human being. And the adjuvant which they used were good. I think many of you know about antibodies and T lymphocytes. Are you all aware of that? Yes? No? Yes, okay. So this adjuvant was a mixture of aluminum hydroxide, which produces very good antibody results, and another compound known as imidazoquinoline, which stimulates the T cells. So it was a consolidated immune response that this company was hoping to achieve by giving this combo adjuvant. So they used different types of adjuvant formulations, different concentrations of the virus antigen. So they gave us 3 formulations. And they also, in our studies, we had what is known as the placebo group. The placebo group is the dummy group. where we will just give them phosphate buffered saline. We will not give them vaccine. So we have, that is the background group, the control group against which we compare the immune response with the 3 formulations of vaccine. So different groups of hamsters were given the 4 formulations. The first is the placebo group, phosphate buffered saline. Group 2, 3, and 4 were those that received different concentrations of the virus antigen and different adjuvant, and 4 groups of hamsters were identified, and they were given the vaccine, the candidate vaccine made by BBIL of the virus we gave them. They were given the vaccine on day 1, day 14, and day 35. 3-dose schedule, and after a gap of about 2 weeks, on the 50th day, the proof of the pudding is in the eating, isn't it? We challenge them with the virus. So when you give them the vaccine in 3 doses, you give a gap, you're giving the gap for antibodies and immune response to kick in and to mount, then you challenge them with the living virus. So we hope that when we do this, the vaccinated animals will have less severe disease, and the placebo or control group will be very sick because they have not received the protection of the vaccine. This was our hope. And what did we find? And of course, once we gave the challenge, we had to by sacrifice animals to look at their lungs, the different organs, to look at the amount of virus these inoculated animals had, and to see what did the vaccine do. Did it actually protect the hamsters when they got the live virus challenge? And what did we see? We saw that if you see, this is the placebo group. This is group 2, 3, and 4, which received different formulations of vaccine. These are images of the lungs of the hamsters. And it takes no imagination to see that the placebo group that really was not protected by the benefit of the vaccine had very hemorrhaging, congested looking lungs on day 2, 1, 3, and 7, versus the 3 groups that received vaccine, their lungs looked much healthier. Okay, so that is what we found. Of course, we did lots of other antibody testing, binding antibody, functional antibody, interleukins. I'm not showing you all of that. That's very scientifically elaborate, but I can take questions later. So hamsters showed a very good response. And these hamsters, we were very picky about the hamsters we got. We got it all the way from the ICMR-National Institute of Nutrition. This came by a vehicle. From there, we had to issue passes because by then there was restricted travel. So as they crossed the state borders, very melodramatic, like you see in the movies, we had to give them passes so that the driver and the technician could come with this big collection of hamsters into the state of Maharashtra, crossing from Telangana to Maharashtra. And every time hamsters were brought into, we had to issue new passes. And every time we did animal experiments, we had to get the okay from the Animal Ethics Committee of the National Institute of Virology, the CPCSCA, which is the national body that prevents cruelty to animals. So we had to submit our proposal, our objective, the final outcome of our proposed outcome of our study. Only after getting approval could we use this many animals for doing what is known as preclinical studies. Clinical means it's with human beings. These are preclinical studies. So then we went on, we ratcheted the level of study to a higher level, what are known as non-human primates. And these are monkeys, as you know, are genetically closer to us, and we decided to do these studies. We had to do these studies in non-human primates. This is the example of a rhesus monkey here. And now maybe we will see monkeys moving around. But during the lockdown, all the monkeys were no more seen in railway stations, in mandirs, in melas, or wherever, tourist attraction spots. Because human beings were all housebound. These monkeys could not get food, so they returned into the jungle. They went right into the depths of the jungle. So we had to capture these monkeys wild caught monkeys. So again, we went through the Animal Ethics Committee, the CPCSA approval, the Conservator of Forests approval, because these are national studies and we cannot take any shortcuts. Approval was given in a matter of 24 hours, normally which would take weeks. Approval was given within 24 hours, and after getting the approval, my scientific colleagues went with the permission letters and approvals in vehicles to capture monkeys, and they went telling me, we will be back in 3 to 4 days. But the monkeys were not to be seen anywhere where they thought they would see monkeys. They came back after 20 days. They almost went so deep into the state of Maharashtra, closer to the Telangana border. In the Nanded and Nagpur forest divisions, they got the monkeys and they returned, and then we had to quarantine them, and then began our studies with the monkeys. Again, the 3 formulations of the virus vaccine were given to monkeys. Monkeys followed a 2-dose vaccination schedule, just like we have got a 2-dose vaccination schedule. And after 2 weeks, again they were challenged with live virus. They were followed up after the live virus challenge, again taking blood, nasal swab, throat swab, rectal swab, fluid from the lungs, taking tissues, urine, stool specimen, and what have you. We even did X-rays on the monkeys. And what did we find? Okay, this is an example of the technician handling the monkeys. I don't know whether you can spot the monkey in its cage. Each of the monkeys were in individual cages. And what did we find? We found, again, this is only representative. This is the placebo group that did not have the protection of vaccine. They just got phosphate buffered saline. This is representative of one of the vaccine groups. Can you see the difference between the lungs? It's very obvious. So we were very sure that this vaccine was protecting. We started with mice, rats, and rabbits, then came to virus challenge experiments in golden Syrian hamsters, came to non-human primates or rhesus monkeys. All were showing a green signal, and then Bharat Biotech was ready to start their clinical trials. So to tell you a little about clinical trials, so as I had said, any vaccine, not just Covaxin, any vaccine goes through preclinical trials. So you can see the examples of monkeys or other animal models where we evaluate safety, estimate what could be a safe dosaging, and identify whether there were any side effects in the animal model. Then very gingerly, very carefully, clinical trials phase 1 starts, wherein we give, of course, after taking permission from the individual who is willing, it's a voluntary participation in a vaccine trial. We explain to them, and once they sign that they are willing to participate, we do what is known as a phase 1 clinical trial, where the vaccine is measured for its effectiveness and also what would be a safe dosaging. We do the tests in a minimal number of individuals. Phase 1 is done only in a small number of individuals. Phase 2, we expand it, the numbers expand. And in phase 3, we have literally thousands of individuals participating. For Bharat Biotech, over 29,000 individuals participated to confirm, one, the effectiveness of the vaccine, monitor any side effects, and compare the effectiveness against any other standard treatment, whether they were, by taking the vaccine, were they protected and not going down with severe disease. We also did preclinical trials for 2 other companies, Zydus Cadila. This has made the first DNA vaccine for the country. So we did these studies again in rhesus macaques or rhesus monkeys. And we also did another vaccine candidate in hamsters for Serum Institute of India. The results were not looking so good, so that particular vaccine candidate was abandoned. We also did a number of other studies which were important for the nation, namely environmental sampling. I don't know whether you know, when any of us get infected with COVID, though it is a virus that infects the upper respiratory tract, eventually virus is shed in the fecal material or stool of the infected individual. That shedding, virus shedding, happens 3 to 4 weeks also after one gets infected. So if you look at sewage water and you get RT-PCR positivity, you know the area from where that sewage water is being collected, that locality has got COVID activity. That means there is COVID, there are COVID positive individuals in that locality based on from where that sewage water is draining from. So this is a very important strategy known as environmental sampling. It's a surrogate or an indirect marker of measuring COVID activity in any particular locality. This we did in 19 different localities or wards in the city of Mumbai, and we have replicated in about 18, 19 other sites in the country. And now many other government agencies have joined this. There is a big network doing this, and we found that the positivity was seen in the first wave, second wave, and third wave, mirroring the outbreak of clinical cases, actual COVID cases. The sewage positivity was also coming positive. So this is described as a surrogate marker. Of course, you cannot identify individuals, but you can identify the presence of COVID positivity in a locality. So these are examples of our technicians taking samples from manholes and sumps in the city. These are not easy to do, to open up sewers, sumps, and manholes in this in a big city like Mumbai. But our technicians did not, refrain from the call of duty, and they went ahead and did the sampling. Now, the virus has actually been changing with time. This is the wave of variants that have been going across from the start of the pandemic to now. Actually, today in July 2022, it hasn't changed much. This GRA, which it's a classification as per a database known as GISAID, as per Pango classification, which is another way to classify the virus. We are now in the era of what is known as the Omicron. The Omicron variant of concern of SARS-CoV-2 is really flooded the whole globe. To monitor this, I'm very proud to say that ever since we found the first variant of concern, different government institutions in India got together and decided that we need to have this genome surveillance. That means you should be monitoring the genetic sequence of positive cases and to see where the variants are appearing. So with the first variant of concern, which came in December of 2020, we formed a network known as the INSACOG, which is the Indian SARS-CoV-2 Genome Sequencing Consortium. And we started with 10 important national labs. Today we are 52 plus labs, which are, just like our police and our military are always on surveillance. These genomic labs are monitoring positive cases to see what are the new variants that India is encountering of this virus. So we are now in a new era, not just of RT-PCR. We are in the era of genomic surveillance, where we look at a variant, a new variant, and does the variant impact on the diagnostics? Are our RT-PCR picking up the variant? Has it become more virulent, or is it more transmissible? Is it affecting, is it responding to the treatment we are giving? Or is it also escaping the immune response that has been generated following vaccination or following natural infection? So this genomic surveillance is something that is going on. This is new capacity building that we have acquired, not just RT-PCR testing, but genome surveillance capacity has been enhanced very much in our country. And I'm proud to say that, again, National Institute of Virology, apart from isolating the first SARS-CoV-2, growing up the virus for the first time in India way back in March 2020, we have been able to grow all the different variants that have been encountered in India. Today we have in our repository over 400 different strains of the virus that we are able to grow up artificially in those cell culture flasks. What do we know about Omicron? We have heard about the Alpha variant, the Beta. We saw very little of it in the country. All of you know about the Delta variant, which caused the crippling second wave. And today we are in the era of the Omicron variant. What is the Omicron variant in compared to the other variants? In terms of transmissibility, highest. We didn't have the Gamma variant. It was mainly from South America. We didn't have it, but this is global data. Omicron is the most transmissible. With regard to escape from antibodies or immune escape, that means the antibody can, to some extent, not completely, but to some extent, escape the antibodies that vaccinated individuals have. Again, Omicron has the maximum antibody escape as compared to the other major variants that we have seen so far. Now, we all are in the era of, yesterday we heard that the government is going to give free booster doses, third dose. And what we wanted to know was, by giving a booster, does the antibody levels actually increase? So this is just one of our studies where we looked at individuals who had received the 2 doses, what we known as the primary course of the vaccine. We looked at their antibody titers, several individuals, and this was 6 months after receiving the 2 doses of the vaccine. We checked the protection against different variants of the virus. This is the original Wuhan strain, the Delta variant, the Beta variant, and the Omicron variant. 6 months after getting the first 2 doses, this was the titer. When we gave the booster, the antibody titers went up. It was the same for all the variants. Of course, with Omicron, it is the lowest. But this slide actually tells you, by taking the third dose or the booster dose, you are able to actually increase the antibodies in individuals who get this third dose. So that's what this slide tells you. And I would like to summate our fight against this virus during this pandemic. We established testing and detected the first cases. We shipped out RT-PCR reactions. I told you more than 64 lakh. We did training, hand-holding. We validated several hundreds of reagents, including made in India products, and were able to say X number of them were good for use in India. We repatriated Indian nationals from Iran, Italy, and from the Diamond Princess cruise ship. We trained several lab personnel through our training video. We isolated the virus, including the ongoing variants of the virus. We made indigenous antibody screening assays, which were used for national sero surveys and for the convalescent plasma trial. We did antiviral testing, sewage water sampling. We provided live and inactivated virus to companies and to academia, even to make equine antiserum. We studied the immune response against various variants, as I showed in the previous slide. We did preclinical trials for vaccines, not just for BBIL Covaxin, but also for Zydus Cadila and Serum Institute of India. And we continue genome surveillance as of date. So how was this possible? This was singularly possible because of teamwork, teamwork, teamwork. And what I'm showing you here is the algorithm which I made, where all my scientific colleagues were in different verticals. There were some in charge of receipt and coordination of samples, those in charge of testing, those in charge of reporting, 24/7 reporting, those who are actually doing the RT-PCR, those who are doing the antibody screening assays, genome sequencing and characterization, virus culture and antigen preparation, kit development, drug testing and drug repurposing, immunological studies, antiviral studies, and also electron microscopy, where I showed you the electron microscope images. We also had people who were looking at the purchase and procurement of reagents. You know, everything has to be procured as per government norms. Distribution of these reagents, we became a central and a regional depot of sending out all these reagents across the country, even during the lockdown. Overall logistics, training, we had to sign many memoranda of understanding, memoranda of agreement, material transfer agreements. So we had people who are looking into that, engaging with the media, and finally other logistics and safety. Safety, we had a group of physician scientists who were monitoring the health of all 700 staff of our institute. I'm very proud to say our field units did their might. They also did a huge amount of testing and dispensation of kits regionally, and they really pulled weight during this pandemic. So experiences learned from our efforts from SARS-CoV-2. First is a proactive approach. You saw, even without the government of India telling us to start screening people who came with very severe respiratory infection, we started doing dummy tests, as I told you in my very first slide. The mantra is teamwork, interdependence. Never to believe I am self-sufficient. Pool our strengths, be interdependent, and we need to have an attitude of resilience that we will prevail, give of our best so that we can actually be of some benefit to our country from the domain of expertise that we have. Diagnostics and research tools needed to be built on quality, and there was no compromise from our side. Introduction of many made in India reagents, because we were able to validate and say X number were good, X number were not so good. We in fact handheld them and tried to improve their quality so that we had a critical mass of reagents, making India self-reliant. The pandemic enhanced laboratory capacity of many small labs, labs that never even knew what an RT-PCR was. They didn't know what it meant. Were all doing RT-PCR. Despite pressure of time, we did not compromise. There was no scope for shortcuts. There was no margin for error. Continued genome surveillance in real time, finding out now if we say there is a new variant, we should find out, okay, somebody is infected with the new variant of the virus. How is the patient presenting? Is his disease mild? Is it severe? Is he hospitalized? Is he in the ICU? Or unfortunately, has he died? We need to associate the variant with the clinical picture. And that is what we have to do in real time. We cannot talk about variants that we detected in February of this year. Today what we see, how is it presented clinically? And effective surveillance always is a way to monitor continually where there can be portals of entry of new variants into the country, into communities, and also we need to think of those underserved more resource poor areas of our country that need augmentation and hand-holding. India's response in this pandemic, we have expanded our laboratory capacity hugely. We have become self-reliant. You know that we have a COVID reporting portal which ICMR set up. We have made our own vaccine. We are making more indigenous vaccines. We have become a supplier of other vaccines, Covishield, originally made as AstraZeneca by the Oxford Group. The technology was handed over to Serum Institute of India, and Covishield is being made by them. Novavax, Serum Institute of India will make it as Covovax. That will also circulate in our country. And one of the biggest boons to our nation has been the CoVIN app related to the vaccination portal. What is India's vulnerability in terms of any future pandemic? We need to keep that in mind. What makes us vulnerable? Our climatic conditions are very vulnerable to many vector-borne diseases such as malaria, dengue fever, and chikungunya, which we suffer with every time and which peaks following the rainy season that we are right now going through. A high-density livestock population from whom viruses can jump from animals and cross from the animal barrier into the human population. So that is known as a zoonotic event, where a virus normally seen in animals can jump into human beings and cause disease. Also, in our country, we are a bit lax about many things like antibiotics. Antibiotics are sold over the counter. They are not consumed as per the prescribed protocol. They are given even to livestock in a very unregulated manner. And therefore, one of the biggest threats we face in the future is antibiotic or antimicrobial resistance, where many of the antibiotics we use will be rendered ineffective because of inappropriate use. We also live in a part of the world where, as I said, the climate and also a very population dense part of the world. Southeast Asia is very high in population. You know, next year, India will become the most populous country in the world. We also have, right now, relatively low healthcare spending, low expenditure on research that could benefit public health. There is scope for better coordination between central and state governments, and our public health infrastructure needs to be augmented little more. It has been augmented after the second wave, definitely, but we need to augment more. The pandemic has taught us that we need to increase the number of trained healthcare workers. We need to have a number of very well-trained epidemiologists who can forecast through mathematical modeling, right mathematical modeling, say what could a new disease evolve into. We also need to have good amount of well-functioning medical equipment in our hospitals. We need to improve our healthcare even in our rural areas. We need to have better and quicker disease reporting mechanisms, better surveillance in all of our states. We need to also have very good messaging to our public, right messaging, so that people take cognizance of a potential threat. Thank you. And also, they take protective measures if required. This is a picture of many of my scientists. You can see they are not all just men, they are also women here. And this is us giving you our COVID punch, that we will prevail. And as an institute, we are now gearing up. Today, we have a suspected case of monkeypox. We are now building up monkeypox testing capacity. We are going to be giving these reagents to the Southeast Asian countries. WHO has requested us. So I would not very hesitatingly say that the National Institute of Virology is like a civilian army. We never rest. Today it is COVID. Tomorrow it could be anything else. So with that, thank you for your patient listening. These are both our campuses. This is the new campus, a newer campus I should say, not so new. And this is the old original campus set up in 1952. I mainly work out of this campus. I go to this campus once in a way, but my main office is here. And this is the iconic Biosafety Level 4, where the most dangerous of pathogens are handled and where all our animal and preclinical trials were done for India's first indigenous vaccine. Thank you once again.
Yeah, can we have the lights on, please? Yeah, now, thank you, ma'am, for such an enlightening lecture. So many unknown facts about ICMR and NIV came, like, I never knew, and I have been fortunate enough to visit NIV, but still, it's an eye-opener for me. So I believe that there might be some questions after these deliberations. If you have some questions, our team is there with microphone. Please stand up so that we can see and we can accommodate few questions.
I want to tell you there is nothing called a stupid question, only a stupid answer. So please ask. And when there are no questions, you know, there are only two inferences. Either you understood everything or you didn't understand anything. And I really hope it is somewhere in between at least, but not definitely the second situation. So I see a young, yes?
Yeah, I wanted to ask, why did you use horses for the testing?
So as I mentioned, horses are a known animal model when you want to make vast amounts of antibody. As I told you about the anti-snake venom, earlier anti-diphtheric serum, diphtheria, you know, antibodies against diphtheria, which is a bacterium, anti-rabies serum. All this was done earlier in horses. This concept is not new, okay? So they were adapting it to SARS-CoV-2. Anti-snake venom is a very important source of ready-made antibody against snake venom, which gets in if you have a snake bite. It's life-saving. So that's why companies came forward to make the horse anti-serum.
Thank you. Good afternoon, ma'am. Ma'am, this is Poonam
Yes.
Pakira from ABSS. And my question to you that after getting the booster dose, how long will the germs last, and does this process need to be repeated?
That's a very, very good question. I mean, I think I'm speaking to a PhD scholar.
No, ma'am, a class 11 student.
No, I know that, but the level of your question is at that level. So yes, unfortunately, this group of coronaviruses do not give very long-lasting immunity. There are viruses like measles virus, for instance. You take 2 doses in early childhood, you are protected for life. But coronaviruses, and this virus comes from a family known as Coronaviridae. Thousands and thousands of relatives in this family in Coronaviridae. Their antibody response or duration of antibody response is short-lived. So the antibodies start declining in about 4 to 6 months. So of course some countries have gone gung-ho. They have given the 3rd, 4th, 5th dose, you know, because the richer countries have just gone really berserk. They keep giving them randomly as many doses. Definitely the 3rd dose or the precautionary dose, what India likes to call the booster dose, is to be given. Antibody titers will definitely be strong and abounding, as I showed you in that slide. It was taken 1 month after the booster dose. But we will have to watch and wait, you know, after 6 months or so, what those antibody titers will be. And a decision will be made whether individuals need to take maybe annual boosters. We don't know. There is no recommendation yet, but this is speculative at this point. So to sum up your question, Yes, antibody titers start declining after about 6 months. Whether we would need subsequent boosters, that needs to be studied and recommendations will be given.
Ma'am, can it be immune?
Sorry?
Can it be naturally immune?
You're talking about after natural infection?
Yes, ma'am.
Even after natural infection, antibody titers wane. In fact, to give you an example, some cousins of SARS-CoV-2, when I say cousins, belonging to the same family of coronaviruses, they cause very mild respiratory illness in young children, even in human beings. When we have a cough and cold, we say we got the flu. But unless we test, it could be a whole slew of different respiratory viruses. So these cousins, coronaviruses, if I were to get infected today, I could pick up the same infection after 8 months because the antibody titers decline. So that is natural infection, and 8 months later, I'm again vulnerable to pick up the infection. So even after natural infection, antibody titers are not as long-lived as some other viruses. If you look at, say, measles virus, mumps virus, rubella virus, hepatitis A virus, hepatitis B virus, all of them give long-lived immunity. But this group of viruses give a relatively shorter duration of immunity. So scientists are looking at that, and believe me, India is not in a bad position. We have very esteemed scientists who are studying this phenomena, and guidelines will come along in due course. Okay?
Thank you, ma'am. Thank you so much.
Yes.
Respected ma'am, thank you for your speech today. I had a question with regards to the future and the aim of for vaccine development. Like, what do you think, like, in your opinion, would be the thing that vaccine development and the virus, COVID-19 virus, has to focus on? Like, what would be the main aim?
So that's another PhD level question. So it's a really good question because that's what I'm going to be talking about tomorrow and to another group. And what we are seeing for the future is that we probably need to change our formulation of the vaccine slowly in keeping with the variants that are emerging. So what some groups, big companies like Pfizer and Moderna are doing, they have what is known as a bivalent vaccine. That means it has the original strain, and that vaccine also has equal amounts of the Omicron strain. So the idea is it will give a wider spread or broader immunity, including variants. And they are trying to include adjuvants. Adjuvants are compounds that will enhance the immunity so that the duration of immunity will also increase. Another concept that is coming is a pan-coronavirus vaccine, where you want to put in as many coronaviruses, especially the known bugs, throw it in using genetic engineering so that it broadens your immune response so that you are at least protected against n number of coronaviruses. But coronaviruses, if you count all the animal and human coronaviruses, human are less, animal, there are over 3,000 coronaviruses. So you will pick some candidate viruses that represent different groups and try and put it into a vaccine known as the pan-corona vaccine.
Excuse me, ma'am. I am Aditi Agarwala of ABSS. I have a question. As you stated before, NIV is running tests on monkeypox. Can you state your observations and what are the symptoms and what it is like?
So monkeypox, as you know, is related to the smallpox virus, but it produces an illness which is much milder than the smallpox. So it produces small eruptions anywhere in the body, particularly where you have been exposed. It mainly spreads through very close contact or even aerosols. If you have very close contact, it's not like SARS-CoV-2. Its capacity to spread is much less. So if you have direct contact of skin, or suppose somebody has used a towel or a face towel or something, you have contact with that used clothing or fomites, what we call, then it is with direct contact that you pick up the infection. So these lesions can come anywhere in the body based on where the contact is. Small lesions like eruptions come, which become larger. They eventually become pustular. That means they look like little balls of pus. And finally, they will erupt and form scab-like lesions. So it is not life-threatening unless, again, somebody has medical complications. But those can leave scars in some individuals, can be disfiguring. So luckily, monkeypox, because it doesn't spread like the way SARS-CoV-2 does, will not, I think, cause a pandemic, but it is spreading between individuals who have very close contact.
I have a different question. Is vaccine also having effects on pregnant women?
Yes, so you're coming back to the COVID vaccine. Yes, so India has taken, and the world over, one of the important steps is first they do it in non-pregnant people, then they do it in pregnant animals. You can't experiment with a pregnant woman, but they take pregnant hamsters, pregnant rats, and they see whether it is affecting the fetus which is growing inside the pregnant animal. And then, of course, they have come to human beings, and we have found that the vaccines in use in our country are safe for use in pregnant women. So, in fact, there's a drive by the government of India to encourage pregnant women to take the vaccine, because the risk of going down with the infection is much more than any possible harm to the baby.
Thank you, ma'am.
Good afternoon, ma'am. I am Tanisha Mehta of DPS. Ma'am, after taking booster dose, many are getting infected from COVID again and again. So, what are your opinions about this?
Okay. So, you know, I am so thrilled I'm here, because the questions that are coming from the students are, you know, the kind of questions that come from adults, science scholars, media people, and what have you. And I think my media friends, many of the questions will be answered by the questions they are raising. So I'm glad you asked that question. You remember I spoke to you about immune escape. That means the virus has got these spikes known as spike protein. On top of the spike is known as the receptor binding domain, which locks with the receptor on our cells. Now, you remember I told you about sequencing the entire genome. So when we sequence, we found that the mutations are also happening in the gene that codes for the receptor binding domain. That means one amino acid that was there has now become another amino acid. So what happens is it can affect human beings in two ways. With the change of the amino acid, the virus can become more transmissible, more infectious. Second, with the change of the amino acid in the receptor binding domain, and remember all vaccines are directed against the spike protein which carries the receptor binding domain. We produce antibodies against the receptor binding domain. When the new virus variant comes, the antibody, because it has changed a little, the capacity of the antibody to neutralize that virus is diminished. The binding is less. So the protection from infection is less. But remember, our immunity is not dependent only on antibodies. Our immunity is also dependent on the cell-mediated immune response through the lymphocytes. You know, the helper T lymphocytes, the cytotoxic T lymphocytes, cell-mediated immunity. And because we take the vaccine, we have protection against serious disease, hospitalization, and death. So I would like to say this vaccine is a vaccine against serious disease, hospitalization, and death, but not necessarily against infection. We might still pick up the infection, but it is mild. That is why we are still saying, chalo, vaccination le lo, precautionary dose bhi lena hai. Kyun? Because you will be protected against the serious outcome of disease. You may get infected, but it is mild.
Thank you, ma'am.
Good afternoon, ma'am. I have a question that we have seen that the different vaccines, they are administered following, means between both the doses, there are some gaps. On what basis are these gaps determined? Covishield has a different gap and Covaxin has a different gap.
So your query is why do they have different time intervals? So those time intervals are generated. You saw the gaps we gave for the hamsters, we gave for the monkeys. So based on preclinical trials and also clinical trials, we find that with this particular gap, this formulation gives the best immune response. You understand? So with a formulation, a 1 month gap or a 6 month gap or a 3 month gap is giving a particular immune response. And based on the best and optimal response, we recommend the spacing between the doses. Okay? So it varies with vaccines and the spacing actually will change based on the formulation of the vaccine. And it's all based on studies. It's not empirical. Based on studies, we look at the immune response and recommend the spacing.
And my second question is that
There are many questions. You can take their writing and then they can be sent in a perspective
Sure, sure.
because it will take a lot of time. You can explain it. Should I explain?
Okay, we will, maybe I can take another, I'll take 5 more questions and we'll, rest can be written in. So please go ahead. Did she have any more queries?
yes, ma'am. My second question was that means it was in relation to a previous answer that you gave. Do the fetus will they have innate immunity against this vaccine? Means against this virus?
See, everybody has some level of innate immunity, but you're not going to put it to test. You know, a fetus which is developing, we don't know what exposure to the virus will do because even the immune system is only developing in the developing fetus. A 3-month-old fetus sitting inside the uterus of a pregnant mother is unlikely to be able to counter the virus. Okay? A fully formed fetus which is close to the full-term delivery may have some level of immune response. So developing fetus is a very vulnerable entity. That's why we are very cautious. Pregnant women are the last group we come to when we embark on a new vaccine. And now it has been certified that these vaccines are safe in pregnant women.
Thank you, ma'am.
Thank you, ma'am, for the wonderful speech. You said that our country is the 5th one to do the virus growing. So I just wanted to ask, what exactly, virus growing exactly helps in what? Like, why is it so important?
Okay, growing the virus, as I told you, we are able to generate a lot of virus of our own to make, first, if we have lot of virus, we can inactivate it and make our own antibody testing platforms. Covaxin would not have been possible if we had not grown the virus. We grew the virus artificially, made sure it was a nice, healthy, robustly growing strain, and that isolated virus was given to Covaxin, to BBIL. Otherwise, today Covaxin would not have been possible. To test antiviral tests, you have to grow the virus and give the antiviral drug. Is the antiviral drug being able to kill the virus while it grows? So the adaptations, even the equine anti-sera, we grow up the virus, kill the virus, give it to the companies that wanted to make horse anti-sera. So it is very, cardinal to any of the verticals of therapeutics, vaccinology, diagnostics, very, very important being able to grow the virus.
Good afternoon, ma'am. Good afternoon, ma'am. This is yet another Agrashenian, Swastika Das. I would like to ask about the 22 out of 100 compounds that you mentioned, antiviral claiming compounds. So are they something which we hear about every day, or like they are yet another scientific?
They were all obscure compounds, and just because it showed some activity in cell culture, it cannot be used for human use. It has to again go through animal studies, potency studies, safety studies. They just came and said, our compound has got antiviral properties. So this was an in vitro, on the laboratory bench exercise. There's much more. Just like vaccines, even making antiviral drugs, there is much more to it. So 22 out of them showed some promise, but we did not take it further because that is not our mandate. If we ran after that, we wouldn't have been able to make the vaccine.
Okay, so from here my second question comes that is there any satisfactory explanation about like how we are killing the animals or dissecting the animals for their lungs? So is there any satisfactory explanation for that?
See, animal activists would be very upset to know that we had to sacrifice a proportion of our animals. But the choice is, do you want to go straight to adults, humans?
No, but for our class we think that, but for the animals that
Yes,
would be very
I do not, disagree with you in terms of sacrificing animals is a cruel thing to do. But you know, in every situation, like a pregnant mother, does she want to have the infection or does she prefer the vaccine if it is safe? So life is full of choices. You want to do it directly in human beings, or you want to check out how animals respond to the vaccine and then in a graded manner take it to human beings. You saw phase 1, phase 2, phase 3, then after that you have to get approval from the regulatory agency and then only it can be used in humans.
Yeah.
So, you know, I had lots of activists that came after me. So I told them, the choice is you want to take a new candidate vaccine or you want it to be checked in a graded manner in animal models, established animal models, its benefit first certified, and then take the vaccine, or shall we use you as one of our guinea pigs?
Thank you, ma'am.
Yeah, so I think I've been prompted that this is the last question.
Good afternoon, ma'am. This is Srilekha from DPS Ruby Park. Ma'am, you said in 2002 we had a SARS-CoV-1 outbreak, but I think that did not last for long. But this outbreak is like lasting for years. So what is the difference between both the outbreaks?
Wonderful question again. You know, the difference between SARS-CoV-1, we call it SARS-CoV-1 versus SARS-CoV-2 for clear distinction is, in fact, that had much higher mortality, but its transmissibility was much less. So we had around 848 cases in the whole world. 848 cases, because its transmissibility was less and we could contain the epidemic and transmission ceased. But the R0 or reproduction number or transmissibility of SARS-CoV-2, though its fatality is less than the first SARS, is so much higher, transmissibility is so much higher. That is why it's making its rounds, wave 1, wave 2, wave 3, round and round the globe. So that's why we are still in the grips of the pandemic.
Thank you, ma'am.
Somebody is pleading for the last question, so we'll call that last. Yeah, I think now, though I'm a big, ambassador for young girls, I think I should encourage questions from the young men here.
Yes. Yes, I am Pratik Mukherjee from DPS and...
Hello. Ma'am, my question was, we have seen from the Wuhan virus till the Delta variant, the viciousness of the virus was very high. But why did the mutation from the Delta variant till the Omicron, the viciousness become less and the transmissibility became higher? Ma'am, is there any study that has been made on it? Why did the mutation suddenly change in between but didn't continue this continuous pathway?
If there's any way to get them straight into a PhD program, I think we should enroll them. Okay, so very good question. Remember, any virus's singular aim is to keep infecting fresh human being and hosts. So it's detrimental for the virus though we cannot predict how the mutation will go. But for the virus, it's detrimental if it is very virulent and kills the host. So it will go to a more, usually, it will go to a less virulent, more transmissible. Its only aim is to jump from one person to another, to another, to another. And that is being panned down in history. Delta was more virulent. Of course, also our health infrastructure was much less then. We were caught, really unawares because of the magnitude of that wave. And Omicron has become more transmissible, dodges the immune system, and relatively more innocuous. It's also more innocuous because the antibody positivity or immunity in the community has gone up so much because of massive vaccination and also a number of people getting naturally infected. But the typical trend of a virus is that it likes to innocently go from individual to individual without causing very overt disease, so that it can keep jumping from one individual to another to make more of its own. Okay?
And in between, the hydroxychloroquine treatment was started in Kolkata and many cities in India also. But why didn't it continue? And nowadays, different drugs from US and different countries were just modified for this COVID, SARS-CoV-2.
Repurposed.
Yes, they were repurposed, but they are still not used. Why? Like,
there's no one country that did not make mistakes, including the United States of India probably made the maximum number of mistakes. So we did not have a typically good antiviral for SARS-CoV-2. That's why we went into these different repurposed drugs that are used for something else, but being repurposed in our combat against SARS-CoV-2. So in the absence of a good antiviral agent, we resorted to use of not only, we used lopinavir, hydroxychloroquine, and a number of other drugs, and remdesivir. Today's study says actually remdesivir doesn't help. Remdesivir is also a repurposed drug. In the absence of that, humanity was desperate to have an antidote. We have monoclonal antibodies which are very good, but the virus is going on changing. So the monoclonals also have to slowly change. So the world is gripped with a challenge where it's a moving target and we have to keep pace with it. So we continued in the absence of another good drug. Remember, Trump was upset that we were not giving them enough of hydroxychloroquine. Okay?
Ma'am, I'm sorry, but ma'am, just one last question. Ma'am, you said that for testing vaccines, small amount of virus was injected in animals. Ma'am, how different or similar were they from the symptoms that were shown in human beings?
So they are very small animals that will not necessarily show symptoms like we show, and neither can they complain of symptoms, so usually it is shown in the form of weight loss. And that's why somebody asked, how will we know what's happening inside the lungs? We have to sacrifice the animal. Okay? Weight loss is a typical surrogate. They become sick. They don't feed. That's the way you can assess that an animal is not well. And to know what is happening inside the lungs, is the vaccine protecting? Is it causing the kind of damage that was seen in human beings following a natural infection? We have to sacrifice these animals, examine the lungs. I've not shown you the picture of the cells. We've got cross sections from biopsy. I've not shown you all of that. We can see virus being collected in different cells inside the lungs in the unvaccinated. But in the vaccinated, the lungs looked almost healthy. Microscopic images of the biopsy. Okay, so that is how you are able to ascertain how animals are either vulnerable to the disease, going down with disease, or they are remaining healthy.
Thank you very much, ma'am, and we are really sorry that we cannot take any more questions, ma'am. Please take this. We cannot take any more questions, but yes, if you have questions, you can send us to our email address at Science City, and we may forward it to her, and she may answer it at some other time. Now, in the end, I would request our director, Sri Anurag Kumar sir, and Director Headquarter, Sir Kumar sir, to please come on stage. I would request our director, Sri Anurag Kumar sir, to please deliver her Thanksgiving address.
Wow. It appeared as if we were listening to the story of 1971 war from a general who actually was on the front line. Thank you, ma'am, very much for giving such an inside story of the war that you all were fighting. And thank you very much for protecting all of us from this dangerous virus. I'm sure this lecture has been a very thrilling experience for all of you who are present in this auditorium. I could see that from the kind of questions that were being asked. And I know that everyone who has not asked questions is having some question in his or her mind. You can, as it was mentioned, you can forward those questions to us, and we'll request ma'am to answer those questions through email to you, to all of you. Thank you, all the schools. Thanks to all the schools who have participated in this lecture. I know we had to, deny entry to some of the schools because of the lack of capacity of this auditorium, and we could not actually visualize the kind of response that we were going to receive. So thanks to all those schools who wanted to come but could not come. I would also like to thank the press and media who have come here in large number, and I know that they are also curious to interact with Madam today. I'm not sure whether she has enough time to do that. In the end, I would also like to thank my colleagues who have organized this lecture in such a nice manner. And I would request all the students in the schools to keep visiting us, because we at Science City aim to bring speakers like Dr. Priya to let you know what is going in the labs of India, in what our scientists are doing, for which we are not directly privy to what is happening over there. And we try to bring some of them over here to make you understand that science is not as boring as it appears. Thanks a lot.